students. I am Dr. Preeti Ameta, Associate Professor, Department of Chemistry, SGG Government College, Maswara. In this video, I am telling about the electron transfer proteins. First is cytochrome. Now, what is the structure and function of the electron transfer proteins? Here you can see the general what I am saying in this videos. First is electron transfer protein. What is electron transfer protein? Those proteins which transfer the electrons from one to another body of the cell, part of the cell you can say. So, cytochrome of the cell is called the electron transfer protein. It is also called transport proteins. Why they are transport proteins? Because they transport the electron or electron to the oxygen. They are found in the living cells, plant and the animals both. Now, cytochromes have two or three types. I am telling you here the iron, sulfur and copper metal containing protein. Copper metal is not in your course, only in the cytochrome. Iron, sulfur and P450 is in your M.Sc. SEM3 course. Now, catalytic cycle of the cytochrome P450 is also, I am telling here, oxidation of hydrocarbon. This is the function of the cytochrome P450. Cytochromes are of three types. You here look parted in the three types. First is cytochrome, iron sulfur protein and blue copper protein. Cytochrome have also called heme proteins because they have heme group. Cytochrome 450 is found in this group. Second is iron sulfur protein having iron, Fe and S, both group in this cytochrome. So it is called iron sulfur protein, also called non-heme protein. It doesn't have heme group. It found not in the heme groups. Now third is blue copper proteins. Non-heme proteins, again they are also called non-heme proteins. They are of three types. Plastocyanin having copper 3 oxidation state, azurine copper 2 oxidation state having the geometry trigonal bipyramidal TBP, ascorbic oxidase, and fourth is ceruloplasmin copper 2 state. These are blue copper protein. You just know the name only because it is not in your course. Now iron sulfur proteins are of two types again. They are called non-heme proteins. They are of two types, rubidoxine and ferredoxine. Rubidoxine have iron 3 and sulfur containing group. Only one iron atom is present. FeS4 is general formula, and this sulfur is not inorganic sulfur. Now ferredoxine have iron plus 3 state, more than one iron atom, 2Fe, 2S, or you can say Fe2S2, S4. Second is 3Fe, 4S contain ferredoxine. Third is 4Fe, 4S. This is called bacterial ferredoxine. 2Fe, 2S is FeS2. This is plant ferredoxine, and 3Fe, 4S cubene called cubene. So these are the general formula of different type of ferredoxines, and this is in the, they are just asking in the objective type questions. What is the general formula of cubene? You can write it in 3Fe and 4S. These are the different type of rubidoxine and ferredoxine. Now, what is heme protein? What is called heme proteins? Heme proteins have some general characteristics. They containing iron plus P ring, means porphyrin ring. Cytochrome are heme, means Fe plus P ring both are present in cytochrome. Then it is called heme protein. Act as a electron carrier, electron transfer agent. Third is active site of cytochrome is iron. Fourth is present in chloroplast and mitochondria. Chloroplast means only plant, mitochondria means both human body and plants. Now types of cytochrome is cytochrome A, cytochrome B, cytochrome C. General structure of cytochrome is, just look at the left side of this slide. You have a structural formula of cytochrome having porphyrin ring. This porphyrin ring having 4 small rings, 1, 2, 3, 4 at the corner of the squares, an active iron site. What is the active site of the porphyrin ring? Is iron. Active site means which is in the center. What do you mean by the active site? Which just first react to anything or activate first in this complete porphyrin ring. Now what is third point of this porphyrin ring is single and double bonds are 11 in number. If you count the double and single conjugated double bonds, then they are 11 in number, all the rings. Now fourth is linkage at the side groups. What are the linkages? R1 and R2. Just at the bottom side you see left side R1, right side R2. 4 CH3 groups. 4 CH3 groups are attached in the 4 porphyrin rings. Third one is CH2, CH2, C double bond O. These are two groups just at the top of this complete structure. This is the basic structures of cytochromes. Now, what is the difference between heme A, heme B and heme C on the basis of the structure? We see here that in heme A configuration, R1 and R2 are different. R1 is equal to CH2 double bond CH2, R2 is C18 H30 OH. In heme B, R1 is equal to R2, both are same, CH2 double bond CH2. In heme C, R1 is equal to R2 is equal to CH, CH3 and sulfur in the protein. So you can just differentiate heme A, heme B, heme C on the basis of the R1 and R2. Now heme A is found in the cytochrome A, heme B is found in the cytochrome B, myoglobin, hemoglobin, both of them found in the heme A and heme B. Found, and third one, heme C is found in the cytochrome C. So you can easily define heme A, heme B, heme C are found in the cytochrome A, B and C. Oxidase form of, now we here just look at the point number second, iron is the active site. So it reacts easily, which is in the center of the porphyrin ring. So the oxidized form of the iron is Fe plus 3 and reduced form of the iron is Fe plus 2. What is the reduced form? Gain of electron. If iron plus 3 gain 1 electron, it converts into the Fe plus 2. So these are the two form of the iron present in the cytochromes. Now, what is the how the electron flow and what is the sequence of the electron flowing in the cytochromes? We see here that flow of electron. This electron flow system is in respiratory chain. Cytochrome A is binded to oxygen. What is the bounded group of oxygen? This is again the question of objective type. Now point 3 is cytochrome B first activate, it transfer electron to cytochrome C, it transfer electron to cytochrome A and it gives lastly the electron to the oxygen in the cytochrome chain I am saying. Now fourth is the reduction potential. What are the reduction potentials of cytochrome B, C and A? Cytochrome B have minimum reduction potential, cytochrome C is average and cytochrome A is the maximum electron reduction potential. Now what is the relation of reduction potential to the electron donating tendency? We see here that the electron donating tendency is inversely proportional to reduction potential. If cytochrome B have minimum reduction potential, then it is a good electron donor. And if any type of cytochrome have highest reduction potential, it is bad electron donor. Now good electron donors have the power of donating electrons to the next type of cytochrome which have slightly higher reduction potential to itself. Then again electron transfer to that cytochrome which have higher reduction potential to itself. So cytochrome B have the minimum, cytochrome C have moderate and cytochrome A have highest reduction potential. So the chain of electron transport is cytochrome B to C to A to oxygen. Now cytochrome C. This is again the question of objective type. Why does cytochrome B first donate the electron? Because the reduction potential of the cytochrome B is the minimum. So it has the highest power to giving the electron. We are donating to the electron. Cytochrome A is connecting to oxygen and it accept electron lastly to the in the cytochromic chain. So because it has the highest reduction potential and the power of accepting electron is maximum of cytochrome A. Now देखिए अब what is cytochrome C? Cytochrome C, the structure of cytochrome C is not exactly known, but books have some just simple reaction, simple structure of the cytochrome C. It has coordination number 6. You just see here in the right, the iron is bounded with 1, 2, 3, 4, 5, 6 type of groups having 5 nitrogen. So it has coordination number how much? 6. 4 carbon sites are occupied by nitrogen of porphyrin rings. Nitrogen of porphyrin rings are occupied by 4 carbon sites. Now fifth site is occupied by the nitrogen of histidine, which is amino acid. These 5, so you can say, you see here that the 5 nitrogen are not same, they are different. 4 are from porphyrin ring and 1 is from histidine, which is an amino acid. So the character of this 5th nitrogen is different from these 4 nitrogens. 5th site occupied by nitrogen of histidine. 6th site occupied by sulfur of methionine amino. Again, this is amino acid. This is iron sulfur protein. We can say iron sulfur protein. Cytochrome C, it can't react due to coordination but react indirectly by electron transfer method, because all the 6 sites are saturated, but it is transferring electron to cytochrome B, then cytochrome C transfer it electron to cytochrome B and then cytochrome A. So it is inert to oxygen. No site is vacant. Any ligand. Now first look here that cytochrome C is inert because it accepts electron from cytochrome B. You just see in the previous slide. Cytochrome C is inert. It accepts electron from the cytochrome B. So it is indirectly transferring electron to cytochrome A. Cytochrome C transfer electron to the cytochrome A. It is inert to oxygen. It is not reacting with the oxygen because there is no site is vacant. Any ligand which is very you can say strong ligand, cyanide or COO, are even not attached to cytochrome C. Cyanide and CO, these are very strong ligand. They are also not react or attached with cytochrome C. In next slide, this is cytochrome A. It binds to oxygen and reduce it. In cytochrome A, you see that again this is hexa-coordinated, have 4 nitrogen. 5th site, it binds with oxygen and reduce it. This vacant site, it has a first one vacant site above the iron. This is the last link in electron transfer chain. In the absence of oxygen, 6th site is vacant. The coordination number is 5. If the oxygen is available there, then the site is attached with the coordinated with the oxygen. So in absence of oxygen, 6th site is vacant. Coordination number is 5. Cyanide is not able to bind to hemoglobin in more amount, but it bound to methemoglobin, means metal hemoglobin. In this, iron is in ferric state, +3 state, not in +2 state ferrous state. So here the iron is +3 state. Met hemoglobin means iron is in +3 state. So the cyanide is attached to the +3 state. Met hemoglobin not able to bind with oxygen. Met hemoglobin is only bound to the cyanide and CO, which are very strong ligands. So it is not bound with the oxygen, not behaving like oxygen carrier. Bluish brown color, very less quantity in the blood. Enzyme NADH have methemoglobin reductase. This enzyme is called methemoglobin reductase. Converts. What is the function of this enzyme? Converts methemoglobin back to hemoglobin. So, what is the enzyme present in the system which converts Fe3 to Fe plus 2? Fe plus 3 no oxygen binding, Fe plus 2 have oxygen binding. So Fe plus 3 is harmful for our body, Fe plus 2 is not harmful, it is favorable to the blood. If Fe plus 3 have vacant site which is not bounded with oxygen, that vacant site is attached to the cyanide. We can see in the next slide what is cyanide poisoning. If cyanide bind to the cytochrome A instead of oxygen, because cyanide is the 200 times stronger than the oxygen ligand, so no transportation of oxygen and it creates poisoning. We can say the potassium cyanide is poisonous to our body, human body. So this is the cyanide poisoning. Why it is cyanide? Why it is poisoning? Because oxygen level of the iron decreases. Vacant site attached with the cyanide groups. So what is the treatment of this? We injected with the amyl nitrite or sodium nitrite, amyl nitrite and sodium nitrite in the body. It converts hemoglobin, methemoglobin to this methemoglobin combined with cyanide. What is the function of amyl nitrate? It converts hemoglobin to methemoglobin. Methemoglobin combined with cyanides and then it liberates hemoglobin. Then it attached hemoglobin and attached to the methemoglobin. Cyanide liberates hemoglobin and attached to the methemoglobin. Then this methemoglobin which is attached to the cyanide, it again attached to the that hemoglobin which is liberated from this reaction is attached with the oxygen, and this methemoglobin is excrete out, or it is water soluble. It's soluble in the water and then excrete out from the body. So we from this treatment we can reduce the cyanide poisoning. Next is cytochrome 450. Cytochrome 450 found in the plants and animals and bacteria. It absorbs as 450 nanometer. When nanometer is used means ultraviolet absorption. If we use the unit centimeter inverse, it means IR spectroscopy. So if we write here 450 nanometer means this is the UV absorption of the cytochrome. Now pi to pi star transition have Soret band. Cytochrome 450 have pi to pi star transition with having Soret band. Blue to red shift. Slightly blue to red shift. Now what is the function? It acts as the catalyst for the insertion of oxygen, monooxygen atom to other organic substrates. Here is the reaction. You just see this function. RH plus oxygen, cytochrome P450 gives alcohol ROH. Example, 2HN plus 2 electron. Carbon have one oxygen, one R and another H. It reacts with oxygen, cytochrome P450 gives ROOH. H is replaced here, substituted by OH. Again, C double bond C, oxygen, cytochrome P450. You can hear epoxide is formed. Again, first of oxygen, this is monooxygen group. Second oxygen is going to water. These are two oxygen molecules found is break into the two oxygen, monooxygen. First is in the epoxide and another one is the water, from the water, reduced to water. Now, what is the structure of P450? This is the structure, general structure of P450. You can draw it, elaborate it form. Iron in the center, plus 3 state, having 4 nitrogen, 4 iron rings, Pr, right here. I am writing here. OH2 is the topmost group, H2O, you can say water, and lower is the sulfur, cysteine ring of the sulfur. It react with the oxygen. Iron plus 3, it accept electrons. Iron plus 3 changes to iron plus 2. This is called reduced cytochrome. If iron plus 3 is present in the plus 2 form, then it is called reduced cytochrome. Iron plus 2 plus oxygen, iron plus 3 plus O2 minus. This is superoxide ion. Bond order of plus 2 is 2. Bond order of Fe plus 3 is 1.5. Oxygen, superoxide ion is 1.5. If bond order decreases, stretching frequency IR decreases, because the nu is equal to 1 of 2 pi under root K upon mu. Bond order is K. If K is decreases, nu is decreases. So the stretching frequencies decreases. Cytochrome P450. It is present in the kidney, converts in soluble hydrocarbon to insoluble hydrocarbons to soluble hydrocarbons, and then these hydrocarbons excrete out, dissolve in the water and excrete out from the body. Here we can say, just in the reaction types, insoluble hydrocarbon cytochrome P450, water soluble hydrocarbon comes out from the body. What is the molar mass of the cytochrome P450 is 15,000, and iron plus 3 is in the low spin octahedral compound. So this is all about the cytochromes. In next video we tell about your iron sulfur proteins.